Hello students, welcome to Zoology Classroom. This is L. Mahesh, Assistant Professor of Zoology from Tara Government College, Sangareddy. In today's class, let us know about cell cycle. So what is cell cycle? Cell cycle is a series of events that occur in the cells. So in this series of events, the cell passes through from the time of its birth until it produces its replica or it produces daughter cell. So that means the events from the beginning of a cell's journey till the end of its journey by giving rise to two daughter cells. So all these events will be repeated in the next generation cell. So all these events, they will occur in a cyclical manner. So all these events that occur repeatedly in the generations of cells are called as cell cycle. So, and cell cycle is responsible for the development and growth in all living organisms. If there are no cell divisions, then we cannot imagine the life on this planet Earth. And the daughter cells, okay, the daughter cells that arise due to this cell cycle or cell division, they will be replicas of their parent cells. And in prokaryotic cells, we see that binary fission will be happening in them. So this binary fission also is responsible for this generation of cells in these prokaryotes. In them also cell cycle occurs, but the division is called as binary fission in them. Whereas in eukaryotes, two types of divisions will be there. One is mitosis and another one is meiosis. So what are the phases in cell cycle? Cell cycle comprises of two phases. One is interphase, second one is division phase. So this division phase, as I have already told, is called as binary fission in case of prokaryotes. In eukaryotes, it is of two types. One is mitosis and another one is meiosis. So, and what is interphase? So interphase is the longest phase of the life of a cell. In case of normal human cells, the estimated lifetime of a cell is around 90 hours. In this 90 hours of lifetime, the human cell spends about 89 hours of time in this interphase only. So 99% of time cell spends as a interphase cell, or it spends its time, 99% of time is spent in interphase only. And it is considered as resting phase. Resting phase refers to the rest from the division, but not rest from any other activity. Actually, metabolically, interphase is the very active phase of the cell. Many metabolic activities will be occurring at very high rates during this interphase. And what happens in this interphase is that mRNA synthesis, rRNA synthesis, tRNA synthesis will be done. Chromosome duplication also will be done. Centrioles also will be duplicated. In that way, two centrioles will be formed. Protein synthesis will be occurring at very high rates. And centrospheres of centrioles and microtubules also arise as the two centrioles are formed. And these microtubules, they form arches, and finally spindle apparatus also will start initiating or forming in this interphase only. And this interphase is divided into three substages. They are G1 phase, S phase, and G2 phase. So here it is a diagram of showing all the cyclical events. So here in this thing, you can see that this M is the mitotic phase or division phase, and G1, later S, and G2. So these three are the subphases of interphases. And coming to G1 phase, it is also called as gap 1 phase or growth 1 phase. Gap 1 phase or growth 1 phase. This is first phase in this interphase. So immediately after the mitosis is finished, parent cell will give rise to two daughter cells. The two daughter cells immediately enter into G1 phase in most cases. In some cases, instead of entering into G1 phase, the daughter cell may also enter into G0 stage. So what is G0 stage? We will see at the end of this lecture. So immediately after the formation of new cell, the cell enters into G1 phase. So, and what happens in this G1 phase? In this G1 phase, the cell will be preparing for the S1 phase. So, in this phase, this is the longest phase in this interphase. Mostly, in many cases, some cells they will spend even years of time in this G1 phase itself. So, for example, nerve cells and muscle cells they will remain in the G1 phase for many years of time. And generally, usually in normal cells, the duration of this G1 phase is about 25 to 50% of the total interphase time. And in this G1 phase, almost all amino acids will be produced or synthesized. From these amino acids, protein synthesis will be done during this phase. And these proteins will be utilized for the growth of the cell, for the growth of the number of increase in the number of cell organelles, and all these amino acids will be utilized in protein synthesis. So, and protein synthesis also requires production of mRNA, RNA, and tRNAs. So, all these also will be synthesized during this G1 phase. New cell organelles will be formed by utilizing all these proteins and synthesized lipids. So concentration of cyclin D also gradually increases. So the cyclin D is very, very important one. So we will see the importance of cyclin D in next lecture. So in synthesis of all DNA polymerase enzymes, remaining enzymes required in the DNA replication also will be done during this interphase, during this G1 phase. And formation of cyclin E complex also will be initiated in this G1 phase. The cyclin E is very, very important to enter into S phase from G1 phase. So the cyclin E production also will be initiated in this G1 phase itself. And the G1 phase can be divided into 4 subphases. One is competence phase. It is also called as G1A phase. Second phase is entry phase, G1B phase. Third phase is progression phase, G1C phase. Fourth one is assembly phase, G1D phase. So all these subphases they may be affected by limiting growth factors, nutrient supply, temperature, and additional inhibiting factors. So all these are limiting factors for these subphases in the G1 phase. And in the G1 phase, a restriction point will be there at the end of this G1 phase. So signals from the extracellular growth factors like transducers or like hormones also, they uh, control the events that happen in G1 phase. So usually we know that growth hormone will be released from our gland, which is below the underneath the brain, that is pituitary gland. So these growth hormones through blood circulation, they reach all the cells. On the surface of cells, receptors are there. When these growth hormone binds to the receptors on the surface of cells, internally many activities will be triggered. So all these activities they will result in this G1 phase activities. So G1 phase activities will be initiated once the growth hormone comes in contact with the cell receptors. So as a part of these things, cyclin protein production also will be done. Cyclin dependent kinase enzymes will be there. We will see about the importance of these cyclin dependent kinase enzymes. So all these also will be activated with the increasing concentrations of cyclin D. So cyclin D is very, very important one. This plays a very important role in finishing the events that happened in this G1 phase. We will see about this cyclin D and CDK enzymes in next class. So immediately after this, mitosis or division phase is finished, 2 daughter cells will be done, will be formed. The 2 daughter cells immediately enter into gap 1 phase or growth 1 phase or G1 phase. Once the G1 phase is finished, at the end of this G1 phase, one checkpoint will be there. So after this checkpoint is successfully met, then the cell enters into S phase. So what happens in the S phase? Let us see. So S phase is called as synthesis phase. So what will be synthesized in this thing? In the G1 phase also, see, we have seen that many proteins will be synthesized, lipids will be synthesized, different types of molecules will be synthesized in G1 phase also. But here we have given synthesis phase, S phase term is given for the second subphase of this interphase. So what is synthesized? So here in this second phase, DNA synthesis, in other words, DNA replication will be done. So S phase, in S phase only DNA replication will be done. All the required components for this DNA replication will be synthesized in the G1 phase. So in G1 phase, not only these components required for this S phase, but also other components required for the growth of the cell also will be synthesized in G1 phase. So the events that happen in this S phase will be controlled by cyclin E and cyclin A. Cyclin E and cyclin A. So two cyclins are important for happening the events in this S phase. And respective CDK, cyclin-dependent kinases enzymes also will be there. So all these cyclins will be synthesized, cyclin E and cyclin A. We will see about the cyclins and respective CDK enzymes in next class. So all these will be synthesized. So by phosphorylating and activating proteins and enzymes, they are involved in this DNA synthesis. The cyclins and CDKs, they control the events in S phase. So in this S phase, DNA synthesis will be done. DNA duplication or DNA replication or doubling of chromosome number will be actually done in S phase. So the cell spends 35 to 40% of interphase time in this S phase itself. So, and in this S phase, DNA synthesis will be completed, finished as quickly as possible. Why? Because if the cell is spending much time in this S phase, what may happen due to the exposure to mutagens and some drugs, the nitrogen base pairs may be damaged or destroyed in the DNA. So we know that DNA will not be in open form in the interphase. Okay, in the interphase, DNA will be in chromatin material form. So DNA will be loosened or DNA will be unwind to replicate. So when it is spending much time in this replicating phase, then due to the exposure to mutagens and various chemicals, the DNA may get damaged or some mutations may take place in them, and these mutations may become a very devastating effect. They may show a devastating effect on the life of the cell. So cyclins, when bound with the respective cyclin-dependent kinases enzymes like CDK1, what they will do, they will form maturation promoting factor called MPF. So these maturation promoting factors, they activate other proteins through phosphorylation. When the other proteins are they are activated, then what they will do in turn, they will control the remaining events that has to happen in this S phase. So immediately after the G1 phase is finished, cell enters into S phase. S phase events are controlled by cyclin E and cyclin A. So once all the events are finished in this S phase, then cell enters into G2 phase. So G2 phase is a pre-mitotic phase or pre-division phase. So in G2 phase is also called as gap 2 phase or growth 2 phase. So this is second growth phase. So in this phase, the cell rapidly grows in size and protein synthesis also will be done. Required proteins required for the division, cell division, be it mitosis or meiosis or binary fission in case of in case of prokaryotes, all the required proteins for these divisions are synthesized in this G2 phase. So nucleus also will be increasing in volume. So nucleus size will be increased. So here DNA synthesis will not be done. DNA synthesis will be finished in S phase itself. Here only size of the nucleus will be increased and metabolic activities required for cell division also occur in this phase. mRNA, tRNA, RNA synthesis will be done. Why? Because protein synthesis is supposed to happen in this phase also. So in G2 phase, number of chromosomes will be doubled in S phase itself, not in G2 phase. And all the other cellular components also will be duplicated for the newly going to form daughter cells. Cyclin A and cyclin B complexes are active during this phase. So once all the events are finished in this G2 phase, then at the end of this G2 phase, one checkpoint will be there. That is called checkpoint 2 or G2 checkpoint. So if the cell is successfully meets the requirements of this G2 check phase, then cell enters into division phase, that is M phase. So in the M phase, we know that in the division phase, during the division phase, what happens? Cell undergoes various changes in them. So as a result of these various events, finally, one mother cell give rise to two daughter cells. So in eukaryotes, these divisions are of two types. One is mitosis, which happens in normal cells other than reproductive cells, and another one is meiosis. So in meiosis, meiosis occurs only in gamete forming cells. So what happens in the mitosis and meiosis, we have seen in another videos, we have discussed in the previous classes. So we are not going to discuss about all those events that happen in this mitosis and meiosis. We know that in the mitosis and meiosis, the events that happen in mitosis and meiosis can be divided into two subphases. One is karyokinesis, another one is cytokinesis. So in the karyokinesis, four subphases will be there: prophase, metaphase, anaphase, and telophase. And in the cytokinesis, once the one nuclei is divided into two nuclei, remaining cell organelles in the cell will be equally divided in between the two daughter cells. Thus, two daughter cells will be formed as a result of this division phase or M phase. In case of meiosis, two meiotic divisions will be there, meiosis 1 and meiosis 2. So all those things we have already discussed, so I'm not going to repeat them. So in some cases, what happens, once these divisions are finished, daughter cells may not enter into G1 phase again. Why? Because if the required conditions are not, if the conditions are not suitable for this G1 phase, or if the conditions are not suitable for again this what division to occur, then under such circumstances, cell is allowed to enter into G0 phase. So what is G0 phase? So in G0 phase, it is actually the resting phase is G0 phase. In this phase, cyclin D will be decreased. So usually cyclin D concentration will be increased in G1 phase. When cyclin D concentration is controlled, cell automatically goes into G0 phase. So how the cell is controlled whether to go into G1 phase or G0 phase is by controlling the production of cyclin D. If the cyclin D production is decreased, cell enters into G0 phase. If cyclin D concentration is increased gradually, then cell will enter into G1 phase. So this cyclin D will be controlled with the help of RB protein. So RB protein is another important controlling regulating factor of this cell cycle. We will discuss about this RB protein in next class. So this RB protein will be in hypophosphorylated form usually. So what happens in the cell cycle, this RB protein will be bind to a special protein called E2F. E2F, these are called transcription proteins or elongation factors. So in the transcription, we know that amino acid chain will be increased or polypeptide chain will be increased. So as the new amino acids are keep on coming and forming peptide bonds, the polypeptide chain will grow in size. So for this thing, some special proteins are required. They are called transcription proteins. In them, elongation factors will be there. Elongation factor to it is E2F. So E2F is very, very important during this protein synthesis. So this E2F proteins are bind to RB protein. So when RB protein is not phosphorylated, it is bind to ETF. So and this ETF are very, very important for cyclin A and cyclin E synthesis. So when this E2F is bind to RB protein, then cyclin A and cyclin E are not formed. When cyclin A and cyclin E are not formed, then cell will not enter into next phase. So automatically cell will not go into G1 phase. So when cyclin A and cyclin E are about to form, then only cell goes into G1 phase. From G1 phase, next cell has to enter into S phase. So cyclin A and cyclin E are required in S phase. But when cyclin A and cyclin E are about to synthesize, then only cell enters into G1 phase. If cyclin A and cyclin E are not going to be formed, then cell will not go into G1 phase. Instead it goes into G0 phase. So growth factor stimulation will be stopped. As a result of that, cell enters into G0 phase. So protein synthesis and all these things very rapidly happening events, all those things will be controlled. So they will not occur with uh, fast rates. So these are the events that have happened in G0 phase. So we can see G0 state in our nervous tissues. So once the baby is born, no new neuron cells are formed in the our bodies or in the newborn babies. So all the neuron cells, they will enter into G0 phase. And this happens in this G0 state can be seen in muscle cells also. So once the baby is born, number of muscle cells also will not be increased. Size of the muscle cells will be increased, but number of the muscle cells will not be increased. Why? Because all the muscle cells immediately after the birth, the muscle cells will enter into G0 state. So in the G0 state, no events that occur that are helpful for the cell division. So in some cases, some other normal cells also, due to the lack of some required chemicals or some required nutrients, some normal cells also may enter into G0 state for a fixed period of time. Once the cell receives required components, required materials or nutrients, then it may come back to G1 phase. So till then, temporarily, cell spends its deficit time in the G0 state. So, but usually this will not happen in case of neuron cells and muscle cells. In remaining cells, the cell after entering into G0 phase, it can come back to G1 phase again. So this is about uh, cell cycle. So how the cell cycle is controlled, we will see in next class.